signing in. Let's start. Hello, everyone, and welcome. Thank you for joining us today. My name is Meng, and I'm a volunteer with the ICSA. I'll be your host for today's session. And I wanted to do a quick introduction to the ICSA. We are the International Chinese Statistical Association with over 22 active members. Today's session is a part of our career development series focused on the pharmaceutical industry. Our topic today is part of our technical training focusing on late-stage clinical trial design. If you like to catch up with our past set webinars, feel free to scan the QR codes on the screen or simply search for us online. I'd like to give a quick big thanks to our subcommittee members and all the volunteers who made this section possible. Now it's my honor to introduce our speaker, Dr. Deli Wang. Dr. Wang is a director of stats and a research fellow at IBV. He holds a PhD degree in biostats and trained in medicine at Beijing Medical University. His expertise includes leading drug development for neurodegenerative diseases like Alzheimer's, and he brings years of experience in late-stage clinical trials. Dr. Wang also have published over 70 papers on statistical methods and support clinical research. Before we get started, here are just a couple quick housekeeping items. Please feel free to drop any questions in the QA box. We'll address them at the end of this presentation. Also, the session is being recorded, and we will send out a link afterwards. All right, let's jump in. Please join me in welcoming Dr. Wang. I'll hand it over to him now.
Thank you, Meng. Let me share my screen. Can everyone see my screen?
Yes, I can.
Okay, thank you, Meng. Okay, thank you everyone for coming to this webinar. As Meng said, my name is Deli Wang. I work for IBV right now. The title for my today's talk is main emphasis of statistical work in the pharmaceutical industry. I have a couple of sessions. Today we'll focus on the first section called late stage clinical trial design. So this is my disclaimer. What I said in this talk represents my personal view and opinion. It does not represent my company's position or opinion. So in today, the objective is today's talk is provide some detailed information for late-stage clinical trials design to understand main components for sample size calculation for late-stage clinical trials. I will talk three major topics. One is designs of later-stage clinical trials. The second one is talk about endpoints in clinical trials. Lastly, we'll talk about main things, sample size calculation for later stage clinical trials. Let's go to the first topic, design of later stage clinical trials. Late stage clinical trials usually refers to phase 2, phase 3, or phase 4 trials. Design for late stage clinical trials vary by phases, endpoints, and trial objectives. So here are listed some trials. We have phase 2A, typically called a proof of concept study. Phase 2B, we call the dose ranging study. Phase 3 is we typically call a confirmatory study targeting for market authorization. And we also have phase 3B, which is mainly for support commercial publication and label enhancement. And phase 4 is mainly for post-marketing studies Majority phase 4 studies are mainly for the safety. In my today's talk, I will focus on the first three, phase 2A, 2B, and phase 3. So for phase 2 trials, after phase 1 trials completed, phase 2 trials may be proposed to test the safety and efficacy for treatment in a relatively larger sample size. So the objective of the phase 2 is to demonstrate proof of concept. We typically call it POC, or dose range for therapy. The sample size per arm for phase 2 trial is usually 60 to 80 per arm, but some trials have about 100 per arm for double-blind trial. The sample size depends on the treatment effect assumptions for the trial. Depending on the study objectives, sample size justification for phase 2 could be based on precision of estimation or powered study with assumed larger treatment effect size, or you can adjust the two-sided alpha level to 10% or 20% instead of 5%, which is typical for phase 3 trial. For the phase 2A, we usually have a two-arm trial. So one active dose arm and one control arm is proposed to prove the concept of therapy. Phase 2B is a dose range trial, is to evaluate dose response for efficacy and safety and select a phase 3 dose if the benefit risk is favorable to a new treatment. Phase 2B could include multiple active arms besides the control arm. For example, you could have a 3-arm study, a 4-arm trial, and a 5-arm trial. But the more arms you have, the sample size needed is more. So we saw the phase 2B trials, 3-arm, 4-arm is very is common, but more than 5 arms is not that common in the industry based on my experience. Dose response relationship could be evaluated in a phase 2B trial. Phase 2 trial is usually designed like an exploratory trial, meaning just like learning the dose of the treatment to select the dose for phase 3. But sometimes the phase 2 can be designed as a registration potential study for some indication. For example, if you do the rare disease trial, the patient population is small, and so you can design a phase 2 study as a registration study. If you design that way, it can be used to support regulatory approvals. Here, this is just an example of phase 2A parallel design. So you can see this is schematic. So this trial include a screening period, and then randomization, and then treatment periods include different visit schedules. And after double-blind period, you can have a post-treatment safety follow-up period. Sometimes the interim analysis could be planned for futility, or the interim analysis you can make a program acceleration for next stage. So this is a very typical phase 2A parallel design. This is an example of a schematic for phase 2B parallel design. Similar like phase 2A design, this trial have a screening period, which is typically used to screen a patient, recruit patient, to see if it meet inclusion, exclusion criteria in order to randomize in a trial. Of course, there is randomization and a double-blind treatment period. And for this example I show here, so in this trial, we have 3 active dose, low dose, middle dose, and high dose, and also have a placebo. It's a placebo-controlled double-blind period. And in this trial, you can plan interim analysis. Here, I show example have 2 interim analyses, interim analysis 1 and interim analysis 2. So I will not talk about the interim analysis in this session, but maybe in the future sessions. So all these interim analyses could be planned for futility, meaning stop the trial early, or you select dose or adjust the dose, dropping arm, or accelerate development to the next stage. So after subjects finish the double-blind period, so in this example, it's 24 weeks treatment duration, and then they have 30 days post-safety follow-up. So this is just an example of in terms of duration of safety follow-up. It depends on investigator drug. The half-life of the drug. If the half-life is very longer, this safety follow-up could be longer. So this is just show you an example of how the design schematic looks like for the phase 2B design. So this is another example that's also the phase 2B parallel design with the extension period. So the reason that typically you finish the double-blind period, how are you going to handle the patient they finish the double-blind period? The traditional way we're doing that, we include one period called the extension period, treatment period. So everybody finish the double-blind period, they will continue follow another period of time for safety, for long-term efficacy look. So the purpose of extension period, there are many reasons. One of the reasons is just like help the trial recruitment, because the patient that don't want the participant trial, they randomize in a placebo group. They want to go to get a treatment, so they hesitate to participate in trial if they are chance they randomize the placebo, especially the probability to randomize placebo is 50% or more. So that's not good. So if you have extension period that provide opportunity for those people randomized placebo subjects, they can receive active treatment if the drug work. So you can see from this schematic, it's just like a Euro parallel design. You have a screening period and double-blind period. Here I use example 24 weeks treatment duration. You have 4 arms and all planned 2 interim analysis. In this case, I just use an example for the double-blind period. And after everybody, each patient finish double-blind period, then they continue treatment. For the active, they continue the same dose in the extension period, and the placebo, they can switch to one of the dose in the extension period. So after extension period or double-blind period, if the patient decide not to roll over extension period, they still have a safety follow-up period. So this is just one example, a typical phase 2B parallel design with an extension period. There are many phase 2 designs. The example I give you is very standard parallel design. In oncology trials, the phase 2 trial could be only one single arm. It's very common for the oncology trial because they're looking for the toxicity or the response rate. There are some other phase 2 trial, they can use the external control or synthetic control. To borrow historical data to accelerate drug development. When you use external control, synthetic control, some extensions you have to pay very attention. Is just like we call the comparability of controls with active arms. Typically, we want to randomize the placebo active. They came from the same population. But if you borrow from external trial, external database, look, characteristics of the population will be different from active arms. That will introduce bias. So this is something that we have to evaluate carefully and make sure that external data control has a similar population with the active arm. And the purpose mainly is to minimize potential bias for estimation in a trial. The other another phase 2 designs we call the operational phase 2/3 design or inferential seamless phase 2 phase 3 design. So this is become quite common in the industry. Mainly is for operational, we want to speed up the enrollment speed up the development for a drug, for new drug development. Another big chunk is phase 2 design is adapt design. So there's a lot of adapt design, say group sequential design, and you do the sample-size re-estimation on the interim, and you do the population adaptive design, and you do an endpoints adaptive design, and the dropping arm design, etc. There's many adaptive design in the phase 2 space. And in this talk, I will not going to talk about those designs, but I just want everybody be aware. This adaptive design is become popular, very popular in industry because it's flexible. It can accelerate development to answer the clinical question quicker than traditional standard design. Okay, the next we'll talk about phase 3 design. That's a design part of the last one I will discuss today. For phase 3 trials are typically considered a pivotal trial to support a new drug application in the United States or market authorization application in European countries. So this is mainly for submission. Sample size for phase 3 is typically large enough, so you have a higher probability of success. Phase 3 design is typically powered as 90% power and a strong control of the overall double-blind error rate if you have multiple hypotheses to be tested in a trial for primary and ranked secondary endpoints. Typical assumptions for phase 3 sample size is you need a minimum 90% power. Double-blind error rate should be controlled one-sided 2.5%. And also you need to consider the missing data rate in a phase 3 trial, and make sure you account that missing data rate in a final sample size determination. Power is mainly based on the primary endpoint, and could be based on also key secondary endpoints or combination of primary and secondary endpoints. Depends on the objectives or clinical questions in a trial. Typically, if you have phase 3 trial, you have protocol and SAP. You usually submit to the regulatory agencies to get feedback. And those feedbacks are typically you included in final protocol and SAP because that is for registration for approval. So regulatory agencies have a big say in terms to approve or not. So feedback from regulatory agencies highly recommended to be incorporated for the final protocol and SAP, unless you have a very strong justification that don't follow the recommendation. This slide just show you an example of a phase 3 parallel design. Is a very standard. I think it's very similar like first one, phase 2A design. But the sample size for each arm is quite different. It will be very large for phase 3 because this is targeting for market authorization and drug approval. And just like other trials, you have screening period and then randomization, and you have a double-blind period. A double-blind typically is required for a phase 3 trial, and have post-treatment safety follow-up. You can plan interim analysis and do early stop for efficacy, just like a group sequential design, or you stop for futility if the drug doesn't work in a phase 3 setting. You already have independent DMC, called the Independent Data Monitor Committee, to use to review safety data or efficacy data if you have an interim analysis. For phase 3, typically a strong control of family-wise error rate is required by regulatory agency, meaning that overall double error rate for all hypothesis testing has to, for the primary and ranked secondary endpoint, need to be controlled within two-sided 5% level or one-sided 2.5% level. If interim analysis for efficacy other than the futility is planned, the alpha control for the interim analysis also needed to be considered as well. There are quite a bit of methods available to adjust this kind of scenario if you have interim analysis. and multiple doses or multiple endpoints. Okay, let's move on to the next big section, the endpoints in clinical trial. So the clinical trials, they have many endpoints. The common endpoints for the efficacy could be like a continuous variable, for example, mean change from baseline at a specific time point, or could be a categorical endpoint, for example, the proportion, incidence rate, or response rate at a specific time point. A survival endpoint, an example, time to death or time to event. This is for survival, or it could be a composite endpoint. For example, the combination of two or more endpoints. One example I have here is a joint assessment of function endpoint, is a continuous variable, and a survival endpoint in LS clinical trials. So this is for the efficacy. And for the safety endpoints, you also could have a continuous endpoint. For example, mean change from baseline lab, vital signs, weight, ECG, all those variables. And also our majority safety is a categorical endpoint. For example, AEs, potential clinical significant events, mortality, etc. In a clinical trial, efficacy endpoints typically is ordered by a family. We call the hierarchical way to indicate the importance of endpoints. So typically, the primary endpoint, efficacy endpoint is in a primary endpoint family. For example, mean change from CDR some box score from baseline at week 76 in an early Alzheimer trial. That could be a primary. In an ALS trial, it could be a joint assessment of function and survival endpoints as a primary. Of course, you have primary, you could have a co-primary, two primary endpoints. Typically, most trials have one primary, but sometimes some trials have co-primary because it depends on the indication. In that case, you have to satisfy both primaries as successful, and then your trial can be successful. The secondary efficacy endpoint include other efficacy endpoint that is interested in. And finally, lowest category is exploratory efficacy endpoint. So I just only have one slide for endpoint, just very high level information. So okay, different endpoints. And the last one I would share the sample size determination in clinical trials. So this is a major work for a trial statistician in a pharmaceutical company, because they design a trial and the sample size, how many patients needed for a trial is critical and very important, and resources driven, and a lot of things rely on sample size. So I will spend more time on talk about the sample size determination in the latest clinical trials. So for the sample size calculation, there are many assumptions that you need to consider. First of all, you need to consider what kind of design you have and what kind of endpoints you have. Okay, given you have design and endpoints, so what's the assumed treatment effect for primary efficacy endpoint? And what's the variability of the treatment effect is? And what's the top one error rate you want to control? So typically for phase 3, is a one-sided 2.5% level or two-sided 5% level for phase 3 trial. And what's the power? Usually for phase 3, we target 90% power. Sometimes you can power is 80%. Is possible. You can propose 80%, but usually we target 90%. But from rare disease, maybe 80% is good. And also you need to consider missing data issue, because in all clinical trials, pretty much you have more or less missing data. So you need to account power loss due to the missing data for your sample size calculation. So phase 3 sample size is typically designed, as I mentioned earlier, in 90% power and a strong control of the overall top-line error rate in a trial. If multiple endpoints are included for primary and secondary endpoints. The common assumptions for phase 3 sample size, we need a minimum 90% power. Top-line error rate control be one-sided 2.5%. Missing data should be considered for final sample size number. For phase 2 trial size, depends on the trial design and the study objective. Sometimes estimation of treatment effect or proof of concept is our objectives for phase 2. And then you, based on those objectives, you determine the size. And sometimes the phase 2 trial can be designed, we call the registrational worthy. This means rigorous enough, like a phase 3 trial, so that a potential submission could occur if data turn out to be very positive. This becomes more and more popular for rare disease indications and sometimes for breakthrough therapies that show very promising results in the early trial. So in that case, you probably don't want to lose this opportunity to submit the results to regulatory agency for approval. For phase 1 trials, typically size is very small, but mainly because the phase 1 trial's objective is evaluate safety and tolerability of a therapy. So size also depends on the trial design. So you have always parameters. So how you calculate sample size for trials, later stage trials? There are some tools available to calculate the sample size. So I think a lot of statistics are probably familiar. In clinical trials, we typically use validated commercial computer software. For example, we use Inquiry or Yeast or others to calculate sample size, because those softwares are validated. The numbers come out can be trusted. So sample size calculation for most common design in clinical trials are available in the Yeast and the Inquiry. And some sample calculation procedures or packages are also available in some statistical analysis software, for example, SAS or R. So you can use those to calculate the sample size as well, as long as the numbers make sense from a mathematical calculation, formula calculation. For specific test statistics published in literature, and the calculation is not available in a standard statistical software, computer codes developed by authors could be used to determine the sample size for a trial. It's preferable that codes are validated for accuracy before you use them in a clinical trial design and analysis. Next slide, I just have a snapshot of Yeast for sample size calculation. This is just like the interface of the software. This functionality-wise, Yeast and Inquiry have a lot of common things. Of course, there are some other software available can calculate, but always came from probably the same literature for sample size determination. So Yeast can call R to perform simulation. If the design is not standard. So in Yeast, I found that it's very useful things, multiplicity control. This is the procedures for top-level control procedures are implemented in the Yeast. For example, they implement Bufferoni, Hochberg, Holmes, fallback, parallel keeping procedure, or graphical approaches. Those are very common multiplicity control procedures to control multiple hypothesis testing in clinical trials. So I feel that in my experience, that's a very useful tool, because typically we run our code to do simulation, but they are implemented in Yeast. I think it's very convenient for you, for the statistician to make sure power is good enough if you have multiple hypothesis testing. Okay, so in terms of calculation of sample size, because the formula depends on the endpoints. So this slide I just show you the continuous endpoint of the mean difference superiority trial for 2 arms, parallel design, 1-to-1 randomization ratio. I think a lot of statisticians may be familiar with this formula. You can calculate sample size. The sample size depends on the top 1 error rate, alpha level, and top 2 error rate is the power. The Z1 minus alpha, Z minus beta are quantiles of standard normal distribution, and alpha is top 1 error rate you specify in your protocol for sample size determination. If alpha level is 2.5% level, this Z1 minus alpha is 1.96. For the power, if you power is 90%, this Z1 minus beta will be 1.28. And the delta is a standard deviation, a standardized treatment difference. We typically call the effect size, which is the mean difference between two groups divided by the common standard deviation. So physicians understand this effect size meaning interpretation from a clinical perspective. But if you have two arms have an unbalanced design, one arm have more subjects than the other arm, so you can introduce key parameter k equal to the ratio of N1 versus N2. So you can use this formula to calculate the sample size. I just use this one example to demonstrate how you calculate the sample size for a continuous endpoint. This study I just found from clinicaltrials.gov. I think clinicaltrials.gov is a very useful website. All the clinical trials is required by law to submit the design, and if you have results, submit publish results in clinicaltrials.gov. So it's free, everybody can use it. It's a very good resource for statistician and physician and the trialist to find information of other trials, other company did, other organization did, use those information to design your studies. The example I use is a Riprazole trial study to treat psychosis associated with Alzheimer's disease. So that study is a phase 3 study, randomized, double-blind, placebo-controlled, use of flexible dosing. Double-blind period is 10 weeks. They have extension period is 130 weeks, and is open label of flexible dosing. And after they finish the extension period, the continued treatment is 140 weeks. That's specifically for France. This all information is coming from clinicaltrial.gov. In terms of treatment for this trial, they have a Riprazole acute phase. They use oral templates, either 1 milligram or 5 milligrams. The different time, the dosing is different. That's why they call it flexible dosing. For example, week 1 to 2 use a 2 milligram dose. Weeks 3 to 4 use a 2 to 5 milligram dose. So that's described in the clinicaltrial.gov. And the dosing is for 10 weeks. The placebo acute phase also is, I think the placebo is matched placebo with a drug. So the treatment duration also is 10 weeks. So extension phase, so they have also is a flexible dosing. So this is all information you can find directly from clinicaltrial.gov. Use that NCT code in here. The reason I use this example because they publish the trial data. I want to use this information to demonstrate how you calculate the sample size. So that trial primary endpoint is changed from baseline neuropsychiatric inventory psychosis subscale score at week 10 in acute phase. So this NPI psychosis subscale consists of 2 domains of delusions and hallucinations. Possible total scores range from 0 to 24. So lower score is desirable. So negative change score indicates improvement. So in the treatment group, I think in this trial, the placebo change at week 10 is a minus 5.52 with standard deviation of 6.6. For the active treatment, they get a minus treatment change is minus 6.55 and the standard deviation of 6.5. So based on information design, based on this information, we want to design a phase 3 trial for AD psychosis. Suppose we want to do that. We use this published data. Assume a placebo decline in our new phase 3 trial is similar to this epirazole trial. So this supposedly new compound called XYZ had treatment effect 25% improvement relative to placebo at week 10, meaning that treatment group NPI's psychosis subscale score change is a minus 6.9. with the same standard deviation of 6.6. So this is all the assumption, but these assumptions came from that trial. So you have this assumption, and then you plug in those assumptions in the yeast. You can calculate sample size for the power 90%, and NPI psychosis subscale at 10 weeks, and use one-sided 2.5% level. So the sample size is 603 per arm. This sample size included 20% missing data for phase 3. So here, so suppose we design study schematic just like this. So you have a screening period, you have double blind with 10 weeks treatment, active or placebo, you have 603. Of course, a visit, you can discuss how many visits you have for double blind period, extended period. You can discuss with the clinical team, operational team to make that decision. It's very similar, like, just like earlier we present, but we have this is a design we are going to have for phase 3. So primary efficacy endpoint is NPI psychosis subscale score. And this is just like, I just give you an example. Suppose we design a trial with continuous endpoint, different effect size. Design is still 2 arms, parallel design, 1-to-1 ratio, but the effect size is different from 0.25, 0.3, 0.35, and Y-sided 2.5 level. And this is the power y-axis versus the total sample size in the x-axis. So this plot does not include the missing data in the presentation. But I think in a trial design stage, you can use this tool and generate a lot of curves, different scenarios, and discuss it with the clinical team and finalize and get alignment with them to determine a final sample size for a trial. Okay, let's look for continuous endpoint. And then let's move on to the binary endpoint. I use example is a difference in proportions. So we also have a formula shown on the screen. So if you have unequal randomization, your key parameters are introduced, and you can use this formula. And similarly, Z y minus alpha, Z minus beta is, I mentioned early, is the same definition. But here we have 2 new parameters, P1 and P2. P1 is a proportion for treatment group, and the P2 is a proportion of control group. If you have a balanced design and you introduce a K parameter, which is the ratio of the sample size of 2 arms. Before clinical superiority trial for 2 proportions with equal randomization, 1-to-1, you can also use the below sample size. So only have one parameter P. The P, which is a pooled proportion of 2 groups, you can use that to calculate the sample size with desired power for the binary endpoint. And here I also have an example to demonstrate this how you calculate sample size for binary endpoint. All this example also came from clinicaltrial.gov. It's a NCT, yeah, it's a X-raptor trial because HCRF. This many treated edema associated with brain tumor. So there are 2 treatment groups. One is HCRF, is a human corticotropic releasing factor, or placebo matched HCRF. So it's a double-blind, randomized, parallel placebo-controlled design. So primary efficacy endpoint is a proportion of patients in each treatment group who are responders at week 2, and also who continue to be responders at week 5. So responders were defined as a study patients who demonstrate the following 3 criteria. At least a 50% reduction in dexamethasone use, those related to baseline. The second is overall 10-item neuro examination score unchanged or lower compared to baseline. Last is Cognitive scale score unchanged or increased related to baseline. So details you can go to that clinical.gov to find the details if you are interested in. all this information is from clinicaltrials.gov for this trial. So based on that, that trial already finished. Okay, they published results. Response rate for the treatment group is 62%. Response rate for the placebo is 48%. So the difference is 14%. So given these results, 261 per arm is needed to have 90% power, with one side at 2.5% level. But if you publish the study published in clinicaltrials.gov, they only have 100 subjects per arm. So if you use the 100 subject per arm, that sample size, you calculate the power based on a 14% difference, you only get a 52% power. But you think about it, if the 14% difference between these two groups is clinically meaningful, then that trial is underpowered. But that's from a stat perspective. But the question is if this 40% really clinically meaningful. But if it's not meaningful, so that trial still probably is okay. But this is just let you know that how you use that information from clinicaltrials.gov and clinical input from parameters to calculate the sample size for your study based on a binary endpoint. And this again just a similar like a continuous endpoint. I just use a different assumption, assuming the placebo response rate is 48%, and active you have 60, 70, 80 percent response rate, and the difference will be different. And then you can see the power curve relative to treatment effect, given the sample size you have. So this is something I think this kind of plot is very useful for statistician to interact with the team, especially the clinical development team, to determine the study design and finalize the sample size for the trial. And the last one is the sample size calculation for the survival endpoint. This is the last endpoint I will discuss today. So survival is different from previous continuous and binary endpoint. We use survival typically is a hazard function for time to event endpoint. So this formula I think you can find in a standard textbook. The hazard function is the density function ft divided by survival function st. And the survival is just exponential of integration of the hazard function. And the survival function can be estimated. Typically, we use a Kaplan-Meier estimator. If you observe data, you can use this non-parametric method to estimate the survival curve. So you can see, oncology trial is very common. Every time you see is a Kaplan-Meier curve plot. If there's treatment effect, the two curves are separated clearly. So that's a Kaplan-Meier plot. So this is very common. I think probably the number one basic for survival oncology trial. In terms of sample size calculation for the survival, so this is one formula I found from literature. So here we are testing the difference of two survival curves, lambda 1 hat, lambda 2 hat. Suppose this is hazard for control and the test group, and suppose delta is a superiority or non-inferiority margin, and you can use a formula on the screen to calculate the sample size needed for the survival endpoint. But here I think sigma lambda 1 hat, sigma lambda 2 hat, all those are variance estimated for hazard lambda 1, lambda 2. It's a very similar like a standard I think a binary endpoint. The format of this formula is quite similar. So in terms of in real practice, when you calculate the survival endpoints of the power, input for survival power calculation can be specified with median survival time difference, or use a hazard ratio with a hazard rate for one group. Or you can use a cumulative survival rates difference for one group or hazard ratio. They all can convert to each other if you assume, say, for example, exponential distribution. They are exchangeable. So you just need one hazard ratio, median survival time, whatever information you have, you can calculate the sample size. In my experience, the median survival time and hazard ratio is easier to calculate, to be used. The hazard is kind of, sometimes some challenge if you don't understand completely what it is. But the median survival time and the hazard ratio are commonly used to support survival calculation. The accrued time and overall survival duration can also be included in the power calculation or time to event endpoint. The reason this accrued time and overall study duration is very important because survival, the power on sample size mainly depends on number of events you observe during the study treatment period. So longer follow-up time and study duration, you have more events observed, and then the power will be higher. So that's why the reason that less information can help you determine the sample size and power. So as on the screen, I'll just show you test the survival curve difference, lambda 1, lambda 2. Let's say the estimate hazard rates for the control test group. So total number of required events, you have a 1 minus beta power. You can use the formula on the screen to calculate. So you have a hazard ratio log lambda 1, lambda 2, which is a log of a hazard ratio. And the P1, P2 here on this formula is a proportion of subjects randomized to active and placebo group. So that basically just like related to the ratio of sample size of two groups. And after you have a number of events required for specific power, so you can figure out what's the sample size needed for survival endpoint. Basically, you just use the number of events divided by probability of a pooled event occurrence during the study period, and then you get total sample size. So there's a publication in 1983 by Shanford to discuss about this. Again, I just a similar pattern. I use an example to demonstrate how you calculate the survival endpoints, sample size and power. So I just use amyotrophic lateral sclerosis as an example. So this LS is a serious neurodegenerative disease. And the median survival time is 3 to 5 years since diagnosis. Suppose we want to design a phase 3 trial using time to event endpoint. So below is a design assumption, hazard ratio of 0.7, median survival time in placebo group is 22 months, power is 90% power, one side is 2.5, and randomization ratio is 1:1, maximum study duration of 45 months, and accrued duration 22 months. So missing data at 12 months is 30% for both groups. So all these are assumptions, and then you plug in the yeast, and then you come out the sample size needed for 90% power. You need 834 total. For each arm is 417. So total number of events you need is 331. So that's it. And based on these assumptions, median survival time for estimated group is 31.4 months. So this is, I think this is what I have so far for the survival endpoint sample calculation. And this is my last slide. I just summarize what I presented today. We go through the standard design for late-stage clinical trials and discuss what that means in clinical trials, and discuss about endpoints for clinical trials and the sample size determination for each individual types endpoints, and the key parameters you needed for sample size calculation for different endpoints, continuous, binary, and survival endpoints. I believe this is my last slides, and this is the reference I used to prepare these slides for presentation. And I just pause here. Thank you very much for your attention, and we can open the floor for some questions.
Thank you so much, Dr. Wang, for this such an informative session. I learned a lot today. I hope the audience did the same way. Yeah, this, especially the deep dive in on the sample size calculation. We all know how important it is. So there's a question from Eugene. Says, I once heard from the director of the FDA that only 50% of phase 3 trial are successful. He wondered she wonder your thoughts on why this happens, given the extensive work or effort, money clinical trials put into in previous clinical trials like phase 1 and phase 2 to understand the efficacy and the safety of treatment effects.
Okay, yeah, this is a this is a challenging question, but I think I just share my thought. Uh, yeah, we have. I think for phase three success rate is much better probably than the phase two. Of course, typically we do clinical trials. We learn step by step. If we know at the very beginning of phase one stage, okay, this drug mechanism of action really work, definitely you want to jump to phase three immediately. But a lot of time, the phase one and the phase two data information is not sufficient for the sponsor to make a 100% correct decision. Say, okay, this drug really work. Because clinical trials are actually related to many things. Mechanism of action. I'll just give you an example of Alzheimer's trials. I work for Alzheimer's for many years. Uh, I think in the past 20 years, there's no success. But people are still doing the clinical trial for Alzheimer's because people are trying different mechanisms, how it works. If we try fail, they try fail. If you don't try, you never find a new drug, right? Everybody knows that clinical trials are very expensive, but because we have a patient, we have to find a way to treat a patient, find a new drug. Since then, I think less I think is just like science. We learn a lot of publication in Nature and Science. How many are true? So this is a very I think is not to industry. Is I think it also be a question to academia and also to the regular agency. Those published very beautiful results in the Nature and the Science. Only a very small proportion can convert to real in a clinical trial. Is really true. So that's I think that's the science, how the biology science, how the works based on my understanding. We hit a failure, but we want a success. But the reality is that we don't we are not God, right? We don't know really drug work out. But I think right now, in my experience, we try to kill the ineffective drug as early as possible. If you especially in a phase two, if you found just like two curves just overlap each other, no hope, we just kill. We don't want to go to phase three. But sometimes, the people go to phase three because there's still some hope. For example, they found, okay, their subgroup maybe potentially work. So then they try the subgroup, but end up the subgroup just maybe by chance because that's a kind of post hoc analysis. So this is something that I think everybody facing, and we are trying to do our best to kill the ineffective therapy as early as possible. And phase one, sometimes like I could take example, for example, amyloid treatment. This is for Alzheimer's. That drugs really work. Remove the amyloid plaque in the brain. If you find in phase one, you can test it very quickly with PET. They removed, but the safety is very high. Error rate is very high phase one. If safety is benefit risk is not favorable to this drug, you would like to stop development, even though the drug will work, because amyloid removal therapy already approved by several phase three clinical trials. It work, but it depends on what you need it for. If you develop a new drug, but your drug works, but safety is a big concern, the company cannot make money. They cannot survive because they spend a lot of investment for the development. So the decision are you going to spend more money to do things that will not benefit to the patient. So there's many things that play around here for this phase three success of drug development. I think everybody know that, but I think some just like we it's random. We try endless, but another day probably after many errors you will find one success. If less success, maybe you will benefit to the patient. But I think this is something is not statisticians facing, everybody is facing in the industry. Let's just share my thought. Doesn't represent my company's view, but it's just my thought. Thank you.
Yeah, Eugene also, also says continue to believe that phase three clinical trials should have better chance of succeeding than a fair correlating test
Yes,
that
yes,
corresponding to the 50% regardless sample size calculations.
yes. The sample size calculation, we have enough sample size to have high probability given assumption work, will have high probability or will achieve that. But if you think drug work, but your power sample size is too small, you cannot detect that difference. That's become a big issue. So that's why in a phase 3 trial, we need to make sure power is sufficient.
That's fair. One last, I think we have a question or time for one last question. Says Dr. Wang, do you mind sharing some R packages that can be used for sample size calculations? Thanks.
Yeah, I think I didn't use R for quite a while, but I think there's a lot of R package. You can, for example, group sequential t-test. There's a lot of packages available in R. You can search that in Google. Sample size calculation use R. They'll come out a lot. You of course you can read the manual because there is a lot of package. But I think before you use R, just make sure the numbers come out from R is aligned with the number you use yeast or unquery. But a lot of time they match each other. But sometimes some software, they do not go through the validation process. Numbers maybe is off. But at least the one thing I just want to remind if you use R. I'm not against use R. I use R a lot. Plot analysis and sample size calculation. Typically for our industry, we use commercial software. Yeah.
That's fair. Yeah, I think we're out of time. We have time here. Before we wrap up, so when I close it, please take a moment to answer our 4 survey questions. Would really help us out on improving our future webinars. Thank you, everyone. Thank you so much, Dr. Wang. Looking forward to speak with you soon again. I hope everyone have a great weekend.
Okay, thank you. Thank you, everybody.
Bye.
Thank you.